Hello, today we'll talk about blood and circulation. This figure shows an axolotl, an organism from the group of organisms which is amphibians. It is a salamander native to the shallow ponds in central Mexico. The feathery red appendages jutting out from the head of this albino adult are gills used for gas exchange. We'll talk about a brief description of the open and the closed circulatory system. In an open circulatory system such as that of the grasshopper, you have an hemolymph. This hemolymph surrounds the body tissues and act as a circulatory fluid. So in other words, there is no capillaries. The hemolymph is bathing the internal organ and there are no minute venules. While in closed circulatory system, such as that of an earthworm, there is interstitial fluid surrounding body tissue, and this fluid is distinct from the blood acting as a circulatory fluid. So you can see that the circuit of the blood is closed between the heart and the organs because there is a lot of branching of the veins and arteries to supply the organs. While in an open circulatory system, it is open, there is a bathing of the hemolymph to the internal organ. Let's see the difference of circulatory schemes in vertebrates. In fish, for instance, there is a single circulation. So what means single circulation? You can see that the blood enters the heart only once. So that it's a one circle. So that's why it's single circulation. The oxygen-rich blood is conveyed to organs and then collected back as the oxygen-poor blood and back to the blood, and where there is oxygenation occur in the gill capillaries. While in amphibians, you can see a double circulation where the blood enters the heart for 2 times. So you can see there is a circulation that enters the blood for oxygenation, and there is a circulation that enters blood for distributing the oxygen-rich blood all over the body. In the first circuit, you can see that the blood, which is the oxygen-poor blood, enter the pulmocutaneous circuit to be oxygenated and then collected back to the heart and then circulated to the organs through the systemic capillaries and then recollected back through the atrium to the heart. That's mean the blood enters the heart 2 times. While in mammals, still a double circulations looks the same in basic plan, but instead of the pulmocutaneous circuit, there is a pulmonary circuit and you have a systemic circuit. And we'll discuss the pulmonary as a mammalian circulation in details in the next few slides. We'll talk detailed on the mammalian cardiovascular system. So as we have said before, it have 2 circulation, the pulmonary circulation and the systemic circulation from step 1 to step 11, and we will discuss this in detail. Starting with the pulmonary circuit, to start with the contraction of the right ventricle that pumps blood to the lungs via the pulmonary arteries. As the blood flows through the capillary bed in the left and right lung, step number 3, it loads oxygen and unloads carbon dioxide. The oxygen-rich blood returns from the lungs via the pulmonary veins to 4, which is the left atrium of the heart. Next, the oxygen-rich blood flows into the heart's left ventricle, which pumps oxygen-rich blood out to the body through the systemic circulation. So you can see that the pulmonary circuit starts from 1 to 5. The end of the pulmonary circulation or the pulmonary circuit is oxygen-rich blood that will be conveyed to the systemic circulation. before the systemic circuit, blood leaves left ventricle via the aorta. The aorta have two routes, either descend and ascend. Aorta ascends to the capillary beds in head and arms, and so actually to provide oxygen-rich blood to the head and the brain and arms. And the aorta as well has another branch that descends into abdomen to supply oxygen to arteries leading to the capillary beds of the abdominal organs and legs. Within capillaries, exchange of gas occur, so oxygen to the tissue and carbon dioxide into the blood. Capillaries rejoin after finishing its job, forming venules, which convey blood to veins. The oxygen-poor blood from head, neck, and forelimbs is collected via a large vein which is called superior vena cava. There is another large vein, the inferior vena cava, which collect the blood from the trunk and the hind limb. The two vena cavae empty their blood into the right atrium, from which the O2 poor blood flows again into the left ventricle to start another pulmonary circuit. Looking into the structure of the mammalian heart, the heart is consists of four chambers, a left and right atrium and left and right ventricle. You can see that there is a number of valves. Generally, the valves prevents the backflow of the blood. There is two atrioventricular valves that prevent the backflow of the blood from the ventricle to the atrium, and there is as well a semilunar valve that prevents the backflow of the blood from the pulmonary artery to the right ventricle, and there is another semilunar valve that prevents the backflow of the blood from the aorta to the left ventricle. You can notice that the walls of each chamber, the thickness of the wall is different, with the left ventricle having the maximum thickness of the wall, that's mean more muscle, obviously because the left ventricle is responsible for pumping the blood through to the whole body and the whole systemic circulation. So what's cardiac output? Cardiac output is the amount of blood pumped per minute from the ventricle, while the heart rate is the number of heartbeats per minute. The heart valve, as we said, it prevents the backflow of the blood, while heart murmur is a condition where there is a defect in one or more valves, so that will permit a backflow of the blood, and this eventually will reduce the cardiac output. We'll talk now about the cardiac cycle. At first, we have an atrial and ventricular diastole, and this during relaxation phase, the blood returning from the large vein, which is the vena cava, flows into the atria and then into the ventricle through the AV valves, the atrioventricular valve. So we have atrial diastole and ventricular diastole. First, the blood returning to the atria and then moves to the ventricle, and this process takes about 0.4 seconds. After that, it starts the atrial systole and the ventricular diastole. This is a brief period, very short period, where the atrial contraction forces all blood remaining in the atria into the ventricle. And this, as we said, is very brief, it's around 0.1 second. This is followed by ventricular systole and atrial diastole. During the remainder of the cycle, which takes around 0.3 seconds, the ventricular contraction pumps blood into the large atria through the semilunar valve. So this ventricular will pump blood, either blue or red, through the pulmonary arteries or through the aorta. Keep in mind that during a diastole, blood flow from veins into heart chambers, while during a systole, blood flows from the atria into the ventricle. For an adult human at rest with a heart rate of about 72 beats per minute, one complete cardiac cycle takes around 0.8 seconds. The blood pressure is recorded as two numbers separated by a slash, the systolic pressure over the diastolic pressure, which the average person have 120/70. Continuing talking about the control of heart rhythm, before describing, you need to understand the figure. So the electrical signal will be represented as yellow, and you need to follow the path of this yellow signal to establish a heart rhythm. The muscle that convey the signal, or the specialized muscle involved in controlling this rhythm, are indicated in the figure in orange. Under each step, you can see the corresponding portion of the ECG, the heartgram. The portion of an ECG is highlighted in yellow. So starting, the signal start at a region called SA node. SA node, which is a sinoatrial node, it's a pacemaker. Actually, this node which control the contraction and the rhythm of the contraction of the heart. This SA node generate electrical signal in atria and set a rate of a heart contraction. Soon after that, the signal are conveyed to the AV node, and the AV node relays signal to ventricle and cause the ventricular contraction. After that, the signal is conveyed, the yellow, as we said before, through the bundles of muscle branch past the signal until reaching the heart apex. So now the signal have moved from the atrium to the heart apex. Once reach the heart apex, the signal spread throughout the ventricle through specialized fibers called Purkinje fibers, and soon the heart will contract, releasing the blood or pumping the blood out from the heart to the lung and the organs. So, as we said before, SA node is responsible for controlling the rhythm of the heart. Some people may have a problem in this SA node, so they need to do an implant, which is artificial pacemaker. This artificial pacemaker actually is a medical device that generates electrical impulse delivered by electrode to contract the heart muscle and regulate the electrical conduction system of the heart. So you can see how the patient have this pacemaker installed above the heart and connected by electrodes to the heart to promote the heart contraction. And because it's a pacemaker, natural pacemaker is not functioning, so he need to install a artificial pacemaker. Blood vessel structure and function. So you can see, as you can see, the artery is covered by a lot of layers, including a connective tissue layer, followed by muscles, then the endothelium. This artery can be broken down into arterioles that soon broke down to capillaries, and the capillaries actually is a very narrow vessels. Actually, it's a one cell thickness of red blood cells to allow the passage of the blood to every single cell inside our body. Soon after that, once the oxygen have been exchanged with the carbon dioxide at the organs, the blood is collected by small blood capillaries into venule, larger venule, that soon collected into a vein that is surrounded as well with the same layers of the arteries. But keep in mind that the thickness of the smooth muscle surrounding the artery is much more than the thickness of the smooth muscle surrounding the vein. And as well, the artery is more narrower than the vein. Once the vein collects the blood, and you can see from the top figure how the walls of the thickness of the muscle is very obvious in the vein, in the artery, more than the vein, and the vein is a much more wide. So how blood flow in vein? So you must keep in mind that skeletal muscles contraction squeeze and constrict the veins, and there is a flaps of tissue within veins, which you can see in the figure, which is called a valve. It is act as a one-way valve that keep blood from moving only toward the heart. If you sit or stand too long, the lack of muscular activity may cause your feet to swell, as blood pools in your vein and will never go back to the heart. So generally, movement will induce the skeletal muscle of the leg, will do contraction that will move the blood through the vein up to until reaching the heart, and the valves close and open successively to prevent the backflow of the blood. So we'll talk now about the composition and function of mammalian blood. Generally, the blood consists of two main components, plasma and the blood cells. For plasma, the main component is water, and its function acts as a solvent, while there is as well in the plasma ions and electrolyte, which the main function of these ions and electrolytes is the osmotic balance and the pH buffering, and the regulation of membrane permeability. Plasma as well contain plasma proteins like albumin, immunoglobulins, which is the antibodies, apolipoproteins, and fibrinogen. And the function of each of them, the albumin is for osmotic balance and pH buffering, while the antibodies for defense, while the apolipoproteins for lipid transport, and of course fibrinogen for clotting. There is other substance transported by blood like nutrients and waste product and respiratory gases and hormones. The other component is the cellular elements, which is around 45%. You have white blood cells, which are 5 different types: basophil, lymphocytes, eosinophils, neutrophils, and monocytes. The main function is defense and immunity. You have the platelets, which function for the blood clotting, and you have, of course, red blood cells, which is function for transport of oxygen and some of the carbon dioxide. If you look closely into the types of the white blood cells, you can see that originally they generated in the bone marrow, which are called from stem cells, that give rise to 2 different lineage of cells: the lymphoid stem cell and the myeloid stem cells. From the lymphoid stem cell, you have the B and T lymphocyte, while from the myeloid stem cells, red blood cells and neutrophil, basophil, and eosinophils and monocytes and platelets are produced. We will talk now about a very important process. It's called the blood clotting. So once there is an endothelium had got damaged, the platelets adhere to collagen fibers in connective tissue and release a substance that make nearby platelets stick. The platelets initially will form a plug that provide immediate protection against blood loss, as you can see from the figure. Unless the break is very small, this plug is reinforced by a fibrin clot. So if the scratch is a very small, there will not be a fibrin clot. Generally, what is happening, so once there is endothelium is damaged, the clotting factor from platelets, damaged cell, and plasma, including calcium and vitamin K, this will initiate an enzymatic cascade which will activate prothrombin to be converted into thrombin. This thrombin will activate more enzymatic cascade to add to the pathway, and the thrombin now is able to activate or convert or promote the conversion of fibrinogen into fiber. And you can see here how the red blood cells is coated in threads of fibrin. So to summarize the fibrin clot formation, actually it's the clotting factors released from the clumped platelets or damaged cell are mixed with clotting factor in plasma, forming an enzyme cascade that converts the plasma protein prothrombin into active form, the thrombin. This thrombin will convert inactive fibrinogen to fibrin, and the threads of fibrin will weave into a clot. And this is the process of blood clotting. So we'll talk now about one type of a cardiovascular disease, which is atherosclerosis. Actually, in atherosclerosis, you can see that if you look into a cross-section into the artery, you can see that the lumen of the artery got very reduced due to the presence of plaque. And obviously this might be due to the eating of a lot of fats, which lead to this sort of cardiovascular disease. So how to relieve this plaque or stop from blocking the stream of blood? So you can see if it's not treated, this atherosclerosis, you can see that the region that on the heart that got blocked can lead to diminished oxygen supply to the muscle of the heart. And this part of the heart will be a dead muscle, and the overall output of the heart will be decreased, and the function of the heart will be lowered. So to relieve this kind of condition or disease, physicians use normally a strategy in the surgery where they implant a stent. A stent, as you can see from figure 1, the stent is with a balloon is inserted into the unobstructed artery, as you can see. Once and you can see on the sides of the stent there is a plaque. Once inserted, they got inflated by the balloon inside it. This balloon inflation will expand the stent and will widening the artery. The balloon is then removed, leaving the stent in place, and this will increase the blood flow. So this stent is like a mesh tube and it will expand the artery, and this will lead to increasing the blood flow. In another approach, they sometimes transplant a healthy blood vessel from the chest or limb to bypass the blockage instead of using a stent. Thank you. Hi. Today we'll talk about gas exchange. Respiration is not breathing. All organisms respire. It is the production of ATP from organic molecules. Aerobic respiration requires oxygen. This is where ventilation and gas exchange comes in. Here you can see that the mechanism of gas exchange differs from that of the fish than of the human. In fish, the gas exchange occurs across thin-mouthed surface in the respiratory organs, such as gills, tracheal systems, and the other lungs. While in human, in the respiratory system, air travels through branching tubes to the lungs where gases are exchanged with the blood. And you can see the transport of gases in the human body. There are circulatory systems where the transport of oxygen to the body tissues and returns carbon dioxide to the lung. So the gas exchange in humans involve breathing, transport of gases, and exchange with body fluids or body cells. So you can see here, breathing starts with breathing in oxygen and getting out carbon dioxide. Soon after that, the circulatory system transport of gases by the circulatory system, that soon in the tissues through the capillaries there is exchange of gases with body cells. So oxygen is delivered to the cells and carbon dioxide are taken out. So the gas exchange occurs across a specialized respiratory surface. Gas exchange supplies oxygen for cellular respiration and disposes carbon dioxide. Animals require large mouth respiratory surface for adequate diffusion of gases between cells and the respiratory medium, either air or water. From this simplified figure, you can see that the oxygen internalization to the organism level is through respiratory surface. That soon the oxygen is carried out by the circulatory system to the tissues at the cellular level to do production of ATP. The byproduct of the production of ATP is carbon dioxide, which soon conveyed to the outside via the circulatory system through the respiratory surface. Gills in the aquatic animals. Gills are outfoldings of the body surface specialized for gas exchange. In some invertebrates, like animals which don't have vertebral columns, like insects, gills have a simple shape and distributed over the body. Many segmented worms have flap-like gills extending from each segment of the body, while the gills of clams, crayfish, and other animals are restricted into local body region. So this is an example of the gills of the sea star. You can see that when we take a section through one of the arms of the sea star, you can see that gills are distributed and extending from the body surface. While here, the gills on the marine worm, you can see they are like out projection or folds outside on each segment of the worm, and they represent the respiratory surface. Here you can see as well in scallop, you can see that the gills are located internally between the bivalves of the animal, internally, two pairs. While in crayfish, the gills are localized as well internally inside the shell of the crayfish. Looking more deeper into the gills in aquatic animals, the effectiveness of gas exchange in some gills, including those of fish, is increased by ventilation and counter current flow of blood and water. That's meaning moving of the blood or the water in opposite direction. So you can see in the fish, the gills are covered by a gill cover called operculum, and the water, once swallowed by the fish, it passes over the gills. When we enlarge this part of the gill, you can see that the blood vessels are moving, artery and veins are moving in the axis, and the gill arch are being bathed by the water flow and through the gill filaments. And when we enlarge a part of these gill filaments, you can see that the gill filament is composed of lamellae, which are extensions that is full of capillaries for the blood flow and for the exchange of gases. You can see that the oxygen-rich blood is collected from the lamellae, where the oxygen-poor blood is conveyed to the lamellae for the aim of gas exchange. You can see that the direction of the blood, of the direction of the water flow, which represented by the blue arrows, blue big arrows, is opposite to the direction of the blood, which is represented by the black arrows, black thin arrows. So again, if we simplified this counter current flow of blood and water, you can see that the oxygen rich blood appears here in red, is moving opposite to water flow, which contains oxygen. As you can see, that the percentage of oxygen is decreased upon gas exchange between the water and the blood, until we have oxygen rich blood represented by the red color. Tracheal system in insects. The tracheal system of insects consists of tiny branching tubes that penetrate the body. The tracheal tubes supply oxygen directly to body cells. Here you can see the locust have air sac, which is extension from the tracheal tubes, and this air sac is open to the outside through a spiracle. While in beetles, you can see that as well the same pattern appears with the tracheal system that opens to outside by an opening as well, and there is a lot of respiratory surface. This opening is used for gas exchange. When we enlarge a part of the tracheal system, and you can see the spiracle, and you can see the branching of tracheoles, and as well you can see the air sacs and the body cells is presented by this orange circles. When you enlarge a part of these tracheoles, you can see it's full of mitochondria and fibrils for contraction, and the tracheoles are embedded inside it. Lungs. Spider, land snails, and most terrestrial vertebrates have internal lungs. An amphibian such as frog ventilates its lung by positive pressure breathing, which forces air down the trachea. And you can see from the figure how the air is breathed in and the lungs are enlarged. For a closer look for the respiratory system in mammals consists of branching ducts convey the air to the lungs. Air inhaled through the nostril passes through the pharynx into the trachea, bronchi, and bronchioles and dead-end alveoli, where gas exchange occurs. And you can see that the oxygen-poor blood, which comes from the heart, The vessel is broken down into capillaries inside the alveoli where exchange of gases occur. Soon after that, the oxygen rich blood collecting the blood from the alveoli is conveyed to the heart. So how a mammal breathe? Mammals ventilate their lung by negative pressure breathing, which pulls air into the lung. So the lung, as you can see from the figure, the lung volume during the process of inhalation, the lung volume increase and the rib muscle and diaphragm contract. So gases diffuse down a pressure gradient. What's meaning down a pressure gradient? It means that the air or gas diffuses from a region of a high pressure to a region of lower pressure, lower partial pressure. In the lungs and the tissues, oxygen and carbon dioxide diffuse from where their pressure are higher to where they are lower. On the contrary, during the process of exhalation, while the air is exhaled out, the rib cage gets smaller and the rib muscle relax and the diaphragm as well relax. This leading to the movement of the diaphragm up, um, um, reducing the volume of the rib cage and expelling out the air during the process of exhalation. So how the breathing in human is controlled? Actually, the breathing in human, the main breathing control center is in the brain in the regions called medulla oblongata. This medulla oblongata regulates both the rate and the depth of breathing in response to pH change in the cerebrospinal fluid. The medulla adjusts the breathing rate and depth to match metabolic demands. So you can see here from the figure that nerve signals trigger contraction of the rib muscles and the diaphragm, either need to contraction or relaxation. While the breathing control center responds to the pH of the blood and the cerebrospinal fluid, leading to sending a nerve signal that integrate as well a nerve signal sends the carbon and oxygen levels in the blood through a sensors that in the aorta of the heart. From this diagram, you can see the process how it occurs. So oxygen is inhaled, it passes through the airspace where airspaces in the alveoli where exchange of gases occur. So the oxygen rich blood is collected to heart and then circulated to the rest of the tissue where through the interstitial fluid there is exchange of gas with the tissues. In the tissues, you have a high pressure of carbon dioxide and a low pressure of oxygen. That's why the oxygen tend to move from the vessel to the tissue, while the carbon dioxide tends to move from the high pressure in the tissue to the vessel. Soon after that, the oxygen, the oxygen poor blood is collected by the vessels represented in blue to the heart to again do another round of circulation through the lungs to get oxygenated, and the carbon dioxide is exhaled out. So what is alveoli and what is the structure of alveoli? Actually, the alveoli is a thin elastic walls have a thin elastic walls for stretching, formed from epithelium, so it's a single cell layer. Beneath the epithelium, there is a dense network of capillaries supplied with deoxygenated blood collected from the right ventricle through the pulmonary artery. Normally in humans, there are about 350 million alveoli with a total absorbing surface around 90 meters square. So you can see from this figure A, the structure of a lung, one lung, and then when we enlarge a part of the lung, you can see that the air passage ends in a tiny pockets which is alveoli. Enlarging more the structure of one alveoli, you can see how the blood supply of the alveoli, how the blood is coming from the pulmonary artery, the oxygen poor blood, and soon after oxygenation it is collected to pass through the pulmonary vein. So the protection of the gas exchange, how is the protection of the gas exchange system from pathogen and particles? Pathogens such as bacteria and dust particles are present in the air we breathe in and are dangerous if not removed. There are two types of cells that provide mechanism to help achieve this. The first one we are familiar with is the goblet cells. Goblet cells are found in the epithelial lining of the trachea, bronchi, and some bronchioles. They secrete mucus, which actually a thin film over the internal lining. This sticky liquid traps pathogen and small particles, preventing them from entering alveoli where they could cause infection or physical damage. While the second type of cells is the ciliated cells, also present in the epithelial lining of the respiratory tract. They continually flicking to move mucus secreted by the goblet cells. They continuously flicking upward and away from the lung. That's why we cough actually. We cough actually, so that means cilia are beating up mucus with collecting the substances or dust to trying to expel them out of the respiratory system. When mucus reaches the top of trachea, it passes down the gullet during normal swallowing. So here you can see the structure of the first figure on the left, where the goblet cells is located, and you can see that all of the cells have like a brush border, which actually cilia. These are the section in the trachea. When we enlarge the figure, you can see how the goblet cell differs from the ciliated cells, and how the goblet cell they have like a cavity, the external cavity for the lot of secretion of mucus. So the breathing rate and how the breathing rate is related to the exercise. During exercise, the rate and the depth of breathing oxygen dissolve in blood, so supply more oxygen, so this for the need of rapidly respiring cells, while the carbon dioxide in blood during exercise increase in blood from muscles. This is detected by the brain, which order the intercostal muscles and diaphragm muscle to more contraction and relaxation and more rapidly. This increasing the breathing rate. So by the end, there is a faster removal of carbon dioxide and deeper breathing, which increase the rate of expiration. So what is respiratory pigment? Respiratory pigments are protein that transport oxygen. In almost all vertebrate is a protein called hemoglobin, which is contained in erythrocytes. Like all respiratory pigments, hemoglobin, which actually loading oxygen in the lungs and unloading it in the other part of the body, so actually it's reversibly can reversibly bind to oxygen. The binding of oxygen to one subunit induces the other subunit to bind oxygen with more affinity, and any drop in pH lower the affinity of hemoglobin for oxygen. So you can see from the figure how the hemoglobin consists of four subunits with a heme group. It is got oxygen loaded in lungs and unloads in tissue, and you can see as well, this is a chemical formula of how carbon dioxide is loaded and unloaded to the blood. The human fetus exchange gases with mother's blood. So you can see the human fetus is connected to the blood, to the mother through the umbilical cord, which actually containing the fetal blood vessels. The placenta containing maternal blood vessels and fetal capillaries, and the exchange of gases occur between the maternal blood vessels and the fetal capillaries. So here this slide distinguishes the difference between respiration, gas exchange, and breathing. So respiration actually it's a series of chemical reactions which happen in all living cells in which food is broken down to release energy, usually by combining it with oxygen. While gas exchange, it's the exchange of gas across the respiratory system. surface. For example, oxygen is taken into the body and CO2 is removed from it. Gas exchange also takes place during photosynthesis and respiration of plants, while breathing, mainly due to the muscular movement, which keep the respiratory surface supplied with oxygen. Thank you.